Hello, welcome to the new tutorial presented by the SchrDbb. In this tutorial, we're going to see sickle cell disease. Now, to see sickle cell disease, we're going to use the SchrDbb algorithm of the disease, the differential diagnosis, the endemic disease, the cause, the complication, the background, then down the prognosis. And this diagram is going to correspond to a theoretical diagram of the mechanism of the disease, the pathogenesis of the disease, the pathophysiology of the disease, and then outside you have the risk factors, and then down here you have the follow-up. Okay, that is it for the disease. Now, in this part, we're going to start with the introduction of sickle cell and then the definition of sickle cell. So, what is the definition of sickle cell disease? Is it correct? If they're asking you for this definition of sickle cell disease, the sickle cell disease is an abnormality in the hemoglobin. Is it correct? It's a genetic. It's an autosomal recessive disease, so it's better to say autosomal recessive disease, which results to an abnormality of the hemoglobin. Is it clear? So autosomal recessive disease is also an abnormality of the hemoglobin. Now, in this, we're going to speak about which abnormality are you speaking about in sickle cell disease. Now, to understand the abnormality, you have to actually know the composition of hemoglobin. Is it clear? You should know the composition of hemoglobin. Now, hemoglobin is made up of two major parts. We have the part called the heme. Hemoglobin has two elements. It has heme and then it has globin. Is it clear? So the heme is not abnormal in the case of sickle cell. When you have abnormality in the heme, you are going to have disease called porphyrias. Is it clear? So if you have abnormalities in the heme, you are going to have disease called porphyrias. Now, abnormalities in the globin. When you have abnormalities in the globin chain is divided into two. You have the alpha globin chain, you have the beta globin chain. Now, when you have abnormality in the alpha globin chain, you have things like the alpha thalassemia. Is it clear? And when you have abnormality in the beta globin chain, you have what is called the beta thalassemia. You have the beta thalassemia, and you have also sickle cell disease. Is it clear? You have also sickle cell disease. So that is the abnormality. So in the sickle cell disease, you have abnormality in the beta globin chain. Now you need to understand that when you have this abnormality in the beta globin chain, beta globin abnormality, you have to ask yourself which abnormality in the beta globin is involved. Is it clear? You need to know that every time that you have proteins, is it clear? When you have proteins are made up of amino acids. Is it clear? And about 21 natural amino acids. Is it clear? So there are about 21 natural amino acids that exist in the human body. Is it clear? Now, you need to know that these amino acids are arranged in chains, is it clear, which can be converted into the primary, secondary, tertiary structures of the proteins. Is it clear? Amino acids are arranged in chains in the primary, secondary, and each of these chains have its position. Is it clear? Each of these chains has its position into the primary, secondary, and tertiary, and all that. So this is how the chain may be in the different structure. Now you need to know that in sickle cell disease, what happens is that at the position number 6 of the amino acid, at position 6 of the amino acid, there is a replacement. Normally there's supposed to be a glutamic acid there. Is it clear? So normally there's supposed to be glutamic acid at the position number 6 of the amino acid of the beta globin chain, but there is a replacement by valine. So valine replaces glutamic acid at the position 6 of the amino acid. So that's the abnormality. Is it clear? So that is the most important thing that you have to know in the introduction. Then the next part of the introduction is that sickle cell disease is a recessive and autosomal recessive condition. For the cause, you have to know there's an autosomal recessive condition. Is it clear? Now, you need to know that what usually they are gotten from parents that have sickle cell trait. Is it clear? They are gotten from parents that have sickle cell trait or the allele for the sickle cell. So now let's come back to understand this. You need to know that what if you have a parent AA, is it clear? You need to know that what generally the hemoglobin which is A, it is the adult hemoglobin. Is it clear? The hemoglobin which is S is the sickle hemoglobin, and this hemoglobin composition can be visualized in the electrophoresis that we're going to see in the diagnosis of sickle cell. Is it clear? Now, you need to know that you can have one parent, the male father, having a relationship with the mother. Let's say that the father is A and A, so I'll just put A for the adult, not to put again HB. A for the father, and then the father is also A, and then the mother is completely sickle cell. You see that on pairing all these, all the children are going to be AS. All of them are going to have the trait, but none of them are going to have the child. That's for the first case. In the second case scenario, you can have AS pairing with SS. In this case scenario, you're going to have one is going to be AS, another is going to be AS, another is going to be SS, another is going to be SS. So half of the population is going to be sickle. It is only when you have the two trait that you are going to be sickle cell. Is it clear? So this is another family that is going to pair. Now the next family that can pair, you can have a male and then a female. In this case now, let's say the male is SS and the female is SS. So all the children, is it clear? All children are going to be sicklers. Is it clear? When you have this type. And then the last one now is when you have a male that is AS and then you have a female that is AS also, you're going to have AA, then you are going to have AS, two of them which are AS, and then one of them is going to be SS. So 1/4 of this family can be having children. So that's why having children that are sicklers. That's why every time that you have a sickle cell trait, it is advisable that when you are doing counseling prior to marriage, is it clear? When you have sickle cell trait, sickle cell patients that have sickle cell can get married to the patient that do not have the trait of the sickle cell completely. Is it clear? So that's the best pair here. The next pair is that patients generally, you need to know that every time you have sickle cell, you should marry somebody that should not have a sickle cell trait at all. So that's in genetic counseling. The next element in genetic counseling is that every time that you have every time, I've also forgotten to pair between the AS and the AA. Is it clear? When you pair between AS and AA, you see that in this case you're going to have AA, you're going to have AA, and then you're going to have AS and AS. Is it clear? So half of the children are going to have the sickle cell trait, but none of them are going to have the sickle cell disease. So if you have a trait also, you can marry now somebody that also homozygous. This is in order to make the sickle cell trait completely disappear from the population. Is it clear? So it's going to make sure that the child is not going to be affected. So from here we are finished with the first part of the study, which is the introduction to sickle cell disease.